Okay. So who's the first person, Miguel? Randomly selected names, of course. Oh, Pauline. Emily. A fine choice, Pauline. Of course, everybody a good choice. I wouldn't say, that's terrible. So, Emily, you want to start? Can we start with A? Do you think you know what A is? Yeah, that's the G1 phase. That's the main growth where the cell spends most of its time in G1. Okay, that's the main deal. Main normal thing. Going through mitosis, all that, that's not the normal state. And we saw that on Tuesday because most of your cells were in interphase, right? Right? It was over 50%. Right? And we'll go back into that data in a second. Emily, what about B? Did you get B? Synthesis, yeah, or S phase. So the chromosomes are duplicated, DNA copied. Okay? Everything is there from G1, and now in S phase we're copying the DNA. All right, let's get somebody else. Miguel, who's next on the list? Or Pauline? Who chooses? What's that? Willman. So what would we got? G1, S, what's next? G2, okay? The cell's double checking, it's ready, it knows it's gonna make a copy, it's just kind of double checking, making sure it's ready to go, okay? Like what do they do on the airplane before it takes off? What do the pilots make sure of? What do they do?
Check the fuel.
They check everything. Check we got enough fuel. Are there any warnings going on? Is everything working properly? Do we have everybody? Everything? Because we can't make a mistake once you're in the air, right? You got to be ready to go. That's too late to catch the mistake later. Before they launch a rocket into space, they're doing thousands of double checks on everything. Okay, so that's what the cell is doing before it launches into the cell division process. Which, Willman, what's the next step? If it decides to divide, what do we call that? Well, before cytokinesis, you have mitosis. And I guess, very specifically, mitosis is not dividing the cell. So I want to make that clear. It's not about dividing the cell. It's about dividing something else in particular. What's being divided in mitosis? Anybody? What is actually being divided? Think about what we were looking at on Tuesday. We were focusing on what structures. And if you look in your biosome packet, especially, right there on 128, you can see them in the diagrams, and you can see them, on page 130. What are we focusing on? What are we dividing very carefully? Lucas, what do you think? Any guess? Like, what's being divided? The what? The nuclear material, yes. And what is that specifically? It's the nuclear material, yes. And what's in the nucleus? What do we call those things, those little worm things? I know, you just don't want to say, but just say it. And if it's not this, then that's good. The chromosomes, I heard it. So it's the division of the chromosomes. So mitosis, write that as a note. Division. Oh my gosh. Division of chromosomes. Yes. That's what mitosis is. You're dividing the chromosomes. It's not really about the whole cell. That's something else. Mitosis is division of chromosomes. Making sure it's half and half. That's the trick. Wants to make sure everything's divided half and half. Okay? All right, so then we have mitosis. That's dividing the chromosomes. One more name. Oh, Kimberly. Choose a name. If you choose your own name, you gotta admit it.
It's Nico.
Nico. What is the next phase? Cytokinesis. And that is, wow, my handwriting is not good today. I had the wrong pen. I don't know. Blame the pen. Cytokinesis. And guess what that's a division of? Division of, what does that kind of sound like? Cytokinesis, cytoplasm. Yes, that's division of cytoplasm. And it's separate from mitosis, although in my mind I kind of throw in there together. But that's a very quick process. And remember, I think yesterday, we talked about the difference between cytokinesis, animal cells and plant cells. So keep that in mind. Now, if we saw let me look at this. This is what the result of a flow cytometry test looks like. So you find a lump in any tissue, okay? They wonder if it's cancerous, okay? So they'll take a biopsy, stick a needle in there, suck out some cells, send it to a laboratory, and they'll do flow cytometry with it. And they're looking for abnormalities in this graph. This is a normal graph, what it should look like. And you see most of the cells, can you read that okay? Most of the cells are in G0, G1, so basically G1 phase. So that's interphase. And that makes sense because that's what we saw, right? We saw interphase. And then fewer down here in S, and then a little bump here in G2 through mitosis. All right? So if there's deviation in what they see here, then it's an indication of cancer. Usually shifting down more towards the synthesis in G2 means that there might be abnormalities, could be cancer. Okay? So if you see a lot of G2s, what might be happening with the cell? Miguel, give me a name. Tell me what you said for number 2. Annabelle. Oh, she's not here, so let's get another one. Ashley. So what do you think? What could be going on here? It isn't. It's not. Something's abnormal. It's getting stopped. Because what's it doing at G2? Like, what does it say? Can you read that up there? Continued growth. But then, can somebody read me what it says up there? Kimberly, can you see it? No? It double checks. Yeah, it's double checking, making sure the chromosomes are okay, the DNA is okay. So if it's getting stuck there, what might be the case, Ashley? Well, if you double check, you see something's not right, is it going to continue? No. So it could mean there's abnormalities with the DNA and chromosomes. Something's not quite right. And like what I just mentioned with cancer, there could be some sort of a cancerous cell situation. The DNA is not copied correctly, and the cell is picking up on that and kind of holding things up. So one possibility, chromosomes, I'm gonna try to slow down, are not copied correctly. There's errors. So it could be some issue with DNA replication, which it's not necessarily cancer, it could be some other kind of disease. Or, it could be a sign that there's cancer is present. And the cell is just overwhelmed. With cell division. Because remember, oops, let me rewrite that. Cancer is when you have cells that just don't know when to stop. They're just dividing and they're not doing the normal stops and checks on cell division. Cancer, unchecked cell division, it just won't stop. And I always thought it was hard for me to understand when I was your age how that causes, death and causes your tissues to shut down. Like, what just having more cells? What's the problem with that? Well, what I learned later, the problem is those cells that are being copied don't function normally, and they take resources and space away from cells that are functioning normally. So the cancer eventually crowds out, destroys the normal cells, and then you're left with a bunch of cells that just aren't doing their job. And then your organs can't function. Think about brain. You've heard of brain cancer, right? So cancer grows in the brain. Those cells aren't doing their normal brain activities. Meanwhile, they push out the parts of the brain that need to work, that are working, and it shuts those parts of the brain down. And then that causes death. When parts of your brain stop working, that's obviously a problem, right? And your kidneys, your liver, if those organs get overtaken by cancer, it's going to shut those organs down too, because the cancer cells just aren't doing what they should be doing. And it's just kind of like being in a classroom. We're having a good time. We're having a good time learning, right? Right? What if we have all these students coming in and then doing other crazy stuff? It's hard to focus, right? And that would be like a cancer on trying to learn something. You're like, I can't. It's crazy in here. Or not other students, but maybe a bunch of wild pigs or something coming here. And, oh gosh, there's wild pigs running around. They're crazy. They're kind of scary. And it takes over the normal functioning. Okay. All right. So I shared my cereal with you. Julian, what's your favorite cereal? Oh, Cinnamon Toast Crunch. That's one of my favorites too. Yeah. Ooh, one thing was similar to that, I think. Golden Grams. I like that. That's kind of like that without the cinnamon. Okay, Pauline, what's your favorite?
Frosted Flakes.
Frosted Flakes too. Okay, so I asked you like the Frosted Flakes. Denise, what's your favorite? Cinnamon Toast Crunch. So good. Jasmine. Cocoa Pebbles. I like those Cocoa Pebbles. Cocoa Pebbles. Wait a second. Are those the chocolate Rice Krispies? Yeah, that's good. And there's Cocoa Puffs. Are the bigger ones, right? But I love the Cocoa Pebbles because they get the milk all chocolatey quick. So good. Cocoa Pebbles. Nico, what's your favorite? Cinnamon Toast Crunch. Yeah, America? Oh yeah, we're talking. Okay. Very good. Jacqueline, what's your favorite? Honey Bunch of Oats. With what? Almonds. I just finished a bag of that the other day. That's probably my favorite go-to. Okay. I love the little granola clumps in there, and it's just good. It seems kind of healthy too. It kind of seems the different things. It just seems better. Yeah, Brittany, what's your favorite? Oh, Lucky Charms with the little marshmallows. I think it's all about those little marshmallows. The way they crunch. It's pretty special. Miguel, what about you?
Frosted Flakes.
Frosted Flakes too. Annabelle, what about you?
What, the beer?
Yeah. Put you on the spot, huh? Well, that's even better. Now you don't have to. What's your favorite?
I think Lucky Charms.
Lucky Charms. Okay, okay. Wilman, did I get you? Pebbles? Coca Pebbles. Lucas, did I get you? What'd you get? Cinnamon Toast Crunch? Okay, that's it. All right. Oh, I almost forgot. Next Tuesday, there is an electives fair, and they want to set up some tables for AP Biology. And I would really appreciate it to have some of you there. And it could be just for a couple minutes, or you can stay longer, whatever it is, just to talk about the class, because we need to get these numbers up, okay? Because I really like to get more people in here and experience some AP Biology. All right, so that's next Tuesday at lunch. So I will offer extra credit if you come. So please, by the end of the class period, let me know if you're interested in that. I would really appreciate it. Okay? Now it kind of be awkward out there. So they're going to be 2 rows of. It's going to be out in the square here with different tables, and you're just going to hang out. Probably maybe bring a microscope down or some other things we've worked on. Maybe set up for some yeast cell. Anyway, something from like some of the labs we've done, and just talk. Be there. We'll see how it goes. Okay. Any questions? You've seen the fairs out there before. You kind of get a feeling of how it works. And another thing I want you to make you aware of. A lot of you are juniors, and I'm not sure if you have, if you're interested in doing more AP Science. If you're in ACE, then I think you can take AP Physics for AP Science. So that's an option. What's that?
Or AP Chemistry?
Yeah. Right now there is no AP Chemistry.
What is it going to bring?
Yeah, we haven't. So last year, it was either going to be AP Chemistry, Environmental Science, or Biology. And we kind of had a split. So we decided to do Biology. But if you're interested in Chemistry or AP Environmental Science, make sure you let your counselor know, or when you go to get class for next year. Because I'll be part of the planning and teaching of those 2 classes potentially. Okay? So I would like to see a lot of you again next year and continue your science journey with you. So just keep that in mind. I'll have a table at least for AP Environmental Science. Potentially Chemistry, just to see what the interest is. Okay? All right, so let's go ahead and finish up the lab we did, kind of look at some of the final calculations. So take that out. And I think I gave you a paper, Kimberly. And I think you guys are finished with the first page, right? First page is done. Okay, so at the bottom, what you're going to do is create a pie chart with the percentages. So your highest percent was probably interface, correct? It's over 50% for most of you, maybe 60, 70% potentially. So you're just going to draw a circle, and then you're going to estimate the different percentages. I estimate this to be, I think 63% is more like this. But you're just doing the best you can. Like the way I estimate it, so what percent is this right here? This whole thing, a quarter, is what percent? What percent is 1/4? Huh? 25, right? Now if I take half of that, what percent is that? 12.5, right? If I take half of that, then it is, what, 6.25, around 6. So you kind of, half of that is, 3%. So just use that when you're doing your calculations. And then for number 2, the mitotic index, that's the percent, or the number of cells in any phase of mitosis divided by the total. So you're going to count all of these cells these are all mitotic cells. You're going to count all of these and divide it by the total. So for my example, I had 23 in any phase of mitosis over 63 total, including the interphase. So 23 over 63, that's my mitotic index. What's that, like 0.38, 0.40, something like that. So it'll be some decimal for your mitotic index. And then go ahead and work on these questions with your people at your group. And you can move around if you want to. And if you want to go back to the stations, you can. Go ahead and work on questions 3, 4, 2, 3, and 4 together. And then I'll come back and talk about the rest of it. Okay? All right, any questions? All right, so you can move around and then work on those questions, and I'll come around and see how you're doing.
Can I get another picture?
Dang. Yeah, I have one left right over there.